In the pathology of rheumatoid arthritis, the joint space becomes a localized battleground flooded with pro-inflammatory cytokines, chief among them tumor necrosis factor alpha, or TNF-alpha. To neutralize this threat, clinicians deploy 5 anti-TNF biologic DMARDs, along with an outgroup biologic, abatacept. These drugs all target the exact same cytokine, yet they have completely different dosing schedules, specific contraindications, and distinct profiles of off-target efficacy. The explanation lies in their molecular architecture, specifically how these molecules differ in the Fc region of the drug. We include abatacept because its distinct structural mechanism provides a vital contrast to standard TNF inhibitors. Whether the Fc region is present, truncated, or completely absent dictates critical secondary functions, such as cellular cytotoxicity and receptor cross-linking. These structural variations establish the clinical boundaries for each drug, serving as a primary roadmap for their utility and their limitations. Let us look at the first category on our matrix, the full-length monoclonal antibodies. Infliximab is a chimeric IgG1 monoclonal antibody. 25% of its amino acid structure is murine, meaning mouse-derived. Because of this, it is highly immunogenic. The human immune system recognizes the murine regions as foreign and develops anti-drug antibodies against it. To counter this, infliximab is administered with a DMARD, like methotrexate, to suppress the immune response. Adalimumab and golimumab solve this structurally. They are produced using recombinant DNA technology to be fully humanized, significantly reducing immunogenicity. Despite their different origins, all 3 of these molecules share a defining trait. They possess a full active Fc domain. This full Fc region allows these antibodies to bind directly to Fc gamma receptors located on macrophages and natural killer cells. This binding triggers active cell lysis through 2 specific pathways: antibody-dependent cellular cytotoxicity, or ADCC, and complement-dependent cytotoxicity. Furthermore, having 2 active binding sites allow them to form massive TNF anti-TNF immune complexes, physically cross-linking the Fc gamma receptors on the surface of macrophages. This specific receptor cross-linking forces a downstream metabolic shift, resulting in the potent suppression of interleukin-12 and interleukin-23 secretion. The active Fc region enables these biologics to drive broad-spectrum immunomodulatory responses, specifically targeting the cellular populations that drive the inflammatory cascade. Moving to the next item on the matrix, etanercept represents a structural anomaly. It is not a true antibody. Instead, it is a genetically engineered receptor fusion protein. Its architecture consists of the human TNFR2 receptor's extracellular ligand-binding domain fused to a truncated IgG1 Fc region. This truncated structure limits its binding capacity. Etanercept binds to soluble TNF in a strict 1-to-1 ratio. These chromatography charts map the physical size of the molecular complexes formed. The data shows a large elution peak for massive infliximab complexes, contrasted sharply with the smaller 1-to-1 peak produced by etanercept. Because of this smaller complex, etanercept physically fails to cross-link macrophage Fc gamma receptors. Without this receptor cross-linking, etanercept is unable to inhibit the downstream secretion of IL-12 and IL-23. This missing mechanism explains a long-standing clinical paradox. While both infliximab and etanercept treat rheumatoid arthritis, etanercept routinely fails to induce remission in Crohn's disease. Neutralizing TNF is insufficient for treating granulomatous diseases like Crohn's. The structural capacity to form massive immune complexes is mandatory for efficacy. Certolizumab pegol sits in the next category of our matrix, representing the most extreme structural reduction of the entire group. Its Fc region has been sliced off entirely, leaving only the target-binding F' fragment. To compensate for the short half-life of a bare fragment, it is conjugated to a polyethylene glycol, or PEG, polymer, increasing its serum half-life and reducing dosing frequency. Because the Fc region is missing, certolizumab has 0 Fc effector activity. Its efficacy relies purely on direct TNF neutralization. However, this deficit becomes highly relevant when looking at the biological mechanics of active materno-fetal placental transfer. To cross the placenta from mother to fetus, a molecule must successfully bind to the neonatal Fc receptor, known as FcRn. Because certolizumab completely lacks an Fc region, it is physically incapable of binding to the FcRn. This engineered deficit provides a major clinical advantage. Certolizumab minimizes placental transfer, making it a premier targeted therapy for women of reproductive age managing active rheumatoid arthritis. We arrive at the final drug, abatacept. While frequently grouped with RA biologics in clinical discussions, abatacept is definitively not a TNF inhibitor. The anti-TNFs we have discussed operate downstream, mopping up inflammatory cytokines after they have already been released into the joint space. Abatacept operates upstream as a selective T-cell co-stimulation blocker. Structurally mimicking CTLA-4, it binds tightly to the CD80 and CD86 receptors on an antigen-presenting cell. This binding physically blocks the necessary secondary interaction with a naive T-cell's CD28 receptor. Without this co-stimulatory signal, the initial activation and proliferation of T-cells is halted entirely, before the inflammatory cascade can even begin. Abandoning cytokine targeting to block the pathway upstream provides a vital, structurally distinct alternative for patients when downstream TNF blockade fails or is contraindicated. Reviewing the completed matrix reveals exactly how these biologics differ. The full Fc region of infliximab, adalimumab, and golimumab grants them broad cytotoxicity and the ability to form massive immune complexes. Etanercept's truncated structure restricts it to a 1-to-1 binding ratio, preventing complex formation and limiting its efficacy in Crohn's disease. Certolizumab's total lack of an Fc region prevents placental transfer, and abatacept bypasses the TNF pathway entirely to block T-cell activation at the source. While neutralizing TNF-alpha is the shared primary mechanism of these therapies, their unique structural modifications dictate the secondary effects that define patient outcomes. Precision pharmacology in rheumatoid arthritis requires translating abstract molecular architecture into concrete clinical realities.